Hello and welcome to this e-seminar, which will provide you with an introduction to designing real-time quantitative PCR assays. This e-seminar will introduce you to quantitative real-time PCR and provide guidance and best practice on how to design, optimize and evaluate the performance of custom qPCR assays. It is assumed that you have a working knowledge of PCR. However, if you require further information, please refer to the 2013 publication Good Practice Guide for the Application of Quantitative PCR by Nolan, Huggett and Sanchez, which is free to download at the URL displayed at the bottom of the slide. In the following section, we will provide an introduction to quantitative PCR, which will include a description and the scope of commonly used chemistries, as well as examples of reporter dyes, compatible quenchers, and double-stranded DNA binding dyes. Quantitative PCR is a modification of the conventional PCR technique, which enables the monitoring of DNA amplification in real time. The modification commonly employs fluorescent dye markers. These markers have been designed such that changes in fluorescent output are in direct relation to the PCR reaction progress. Hence, the level of fluorescence detected at the end of each cycle will reflect the amount of amplified product present, and can be used to determine the abundance of the target in the original test sample. The real-time monitoring of changes in DNA concentration during a PCR reaction is commonly achieved through the use of fluorescence-based chemistries. Commonly employed chemistries include double-stranded DNA binding dyes and sequence-specific dual-labeled hydrolysis probes. Double-stranded DNA binding dyes exhibit an enhanced capability to emit fluorescent light on excitation when they are intercalated with double-stranded DNA. With hydrolysis probes, fluorescence is detected as a consequence of the release of a reporter dye molecule from the probe as a result of the enzymatic action of DNA Taq polymerase. As stated previously, a commonly used quantitative PCR chemistry involves the use of double-stranded DNA-binding fluorescent dyes. These dyes exhibit an enhanced level of fluorescence on excitation when intercalated with double-stranded DNA. As a consequence, fluorescence can result not only from the PCR product generated during the reaction, but also from any primer dimers or non-specific PCR products. Additional analytical techniques need to be employed in order to compensate for the presence of non-specific PCR products. An alternative and frequently employed fluorescent chemistry is that of dual-labeled DNA sequence-specific hydrolysis probes. With this class of chemistry, the proximity of a fluorescent reporter molecule to that of a quencher prevents the emission of fluorescence from a probe molecule in free solution. During PCR extension of the primers by Taq DNA polymerase, the reporter is cleaved from the hybridized probe, which contains the quencher, resulting in the emission of fluorescence from the reporter when it is subject to excitation. An example of dual-labeled hydrolysis probe chemistry is the Taq-Man probes, the reaction basics of which are illustrated on the current slide. Molecular beacons are single-stranded oligonucleotides that can form stem and loop structures in solution. The loop contains a DNA probe sequence complementary to that of a target sequence, and the stem is formed by the annealing of complementary arm sequences located either side of the probe sequence. A reporter fluorophore is linked to the 5' end of one arm, and a quencher to the 3' end of the other arm. On hybridization of the probe to the target, a conformational change occurs which results in the separation of the reporter from the quencher, enabling the molecule to fluoresce. Scorpion probes resemble molecular beacons in structure, but with the addition of a covalently linked forward primer at the 5' end of the molecule. During the initial qPCR cycles, The Scorpion primers are extended and become part of the resulting amplicon. During subsequent PCR cycles, the probe sequence anneals to the target, causing separation of the quencher from the reporter and enabling the reporter to fluoresce on excitation. The reaction kinetics for the Scorpion system make for very rapid reaction times. In addition, Scorpions are highly target-specific, which makes them suitable for the detection of single nucleotide polymorphisms. Double-stranded DNA-binding dye and hydrolysis probe-based chemistries share a number of key features, as well as possessing those that are specific to the chemistry. The key difference between the chemistries is that hydrolysis probes can be used to detect specific amplification products, whereas double-stranded DNA binding dyes detect all of the double-stranded DNA molecules present, which can include primer dimers as well as non-specific PCR products. Both chemistries can be employed to detect as well as quantify specific DNA targets. However, hydrolysis probes are more specific and offer a straightforward way to multiplex assays. There are a large number of reporter dyes, quenchers and double-stranded DNA binding dyes, and a limited number of examples are shown here for illustrative purposes. Selection of a suitable dye or quencher will be based on the requirements of the analysis to be performed, but key considerations will include the spectral coverage capability of the real-time PCR instrument to be employed, and the need to avoid spectral overlap if multiplexing assays. The following section provides example guidance, recommendations and best practice for the design of quantitative PCR assays. There are 5 key steps in the development of custom quantitative PCR assays, which are summarized in the flow diagram illustrated here. However, the current e-seminar is aimed at the steps involved in stage 2 of the process and will therefore focus on the selection of an amplicon and the design of primers and probes. An amplicon is the target DNA sequence to which an assay is to be designed. It is likely to be a section of a much larger region of a sequence that has been identified as potentially suitable for discriminating one sample, e.g. plant species, from another. There are a number of recommended criteria for selecting an amplicon. It should be between 75 to 150 bases in length and lack significant secondary structure. Regions of high GC content should be avoided, as the PCR products to be generated should be of approximately 50% GC. The amplicon should also be devoid of regions of repetitive sequences, palindromes and SNPs. The design of primers and probes is a stepwise process. The initial step is to identify a DNA target and then, if possible, to obtain multiple DNA sequences for this region. This will usually require searching multiple DNA databases. Once a sufficient number of DNA sequences have been obtained, they should be aligned using appropriate software, regions of homology and polymorphism identified, and a consensus sequence generated and exported. The exported sequence can then be used in an appropriate software package to design primers and probes based on a number of design criteria that are described on the next slide. The slide illustrates the general criteria that should be applied when designing quantitative PCR primers and probes. For primers, a length of between 20 to 24 bases and a predicted target melting temperature Tm of approximately 60 degrees centigrade is recommended. Primer-based composition should be between 40 to 60% GC, and the difference in melting temperature between primer pairs should not exceed 2 degrees centigrade. For probes, a design length of between 20 to 30 bases is normal, but the theoretical melting temperature should be between 7 to 10 degrees centigrade higher than the average for the primers. This will ensure that the probe hybridizes before the primers and in advance of primer extension by the DNA Taq polymerase. For quantitative applications, the probe should be sited close to the 3' end of the forward primer, but not be overlapping. It is also preferable that there are no more G's than C's in the probe sequence, and that there isn't a G at the 5' end of the probe, as this will cause quenching. In addition to the main design criteria described on the previous slide, there are a number of additional key points to address. Ensure that the 3' ends of the primers are not complementary, as this can promote the formation of primer dimers, and that internal sites of self-complementarity are avoided, as these can lead to secondary structure formation. Runs of 3 or more C's or G's at the 3' ends of the primers should be avoided, as this may promote mispriming. The melting temperature provides an indication of how strongly two strands of DNA will hybridize together. For quantitative PCR primers, the melting temperatures should be approximately 60 degrees centigrade, i.e. 59 degrees centigrade plus or minus 2 degrees centigrade. For the probe, a melting temperature of between 7 to 10 degrees centigrade greater than that for the average of the primer pair is generally recommended. A number of methods have been proposed for estimating the melting temperatures of oligonucleotides. A widely employed method is based on the Wallace equation, which allocates 2 degrees centigrade for every A and T base present, and 4 degrees centigrade for every G and C base. However, most primer design software will perform the necessary melting temperature calculations. It is generally recommended that appropriate software be employed to assist with the design of quantitative PCR primers and probes. A number of oligonucleotide design software products are currently available, which offer a broad range of options for oligonucleotide design and evaluation. Examples of non-commercial primer and probe design software are illustrated on the current slide. These include Primer Blast from the NCBI, Primo 3.4 from Chang Bioscience, and Oligo Analyzer 3.1 from Integrated DNA Technologies. A broad range of commercial oligonucleotide design software packages are now available, 3 of which are illustrated on the current slide. Each of the illustrated products provides a comprehensive primer and probe design analysis package, the use of which have been cited in the scientific literature. Primer Express and Vector NTI Express are both distributed by Thermo Fisher Scientific, while Oligo is currently available from Molecular Biology Insights. The current slide illustrates the oligonucleotide input page of the web-based interface for the NCBI's Open Access Oligonucleotide Design Tool, Primer Blast, as accessed on the 3rd of July 2018. The DNA sequence for the target region can be copied and pasted into the input dialog box in FASTA format. Alternatively, existing sequence files can be linked and uploaded to the software directly. The software enables a number of primer design parameters to be specified, including product size, melting temperatures, primers and probe, as well as the relative primer positions within the target sequence. The software provides a graphical output, which enables the user to visualize the relative positions of the different primer designs. It also provides the DNA sequences for the primers and probe, as well as their primer lengths, melting temperatures, percentage GC content, and relative start and stop positions within the DNA target region. The likelihood of primer self-complementary structures occurring for both the 3' and 5' ends of the primers is also determined. This is evaluated by a numerical score, for which the larger the value appended to the design, the greater the chance of a self-complementary event. Interpretation of the Primer Blast output is summarized on the current slide. Primer and probe sequences are presented in the 5' to 3' orientation. The individual primer and probe characteristics provided include the length in terms of nucleotides, the relative start and stop positions within the input sequence, the individual melting temperatures of the primers and probe, the primer and probe GC content, and the degree of primer self-complementarity that could be expected. Evaluating primers and probe for potential cross-reactivity with non-target organisms is recommended. These types of evaluation screen for similarities between the design sequences and entries contained in pre-designated DNA sequence databases, e.g. GenBank. As such, the level of determined specificity is only as accurate as the sequence annotation available, and in practice, experimental evaluation of the designed assays should be carried out using appropriate reference materials. Important points to consider when performing in silico specificity evaluation are ensuring the correct name for the target is employed, that other species likely to be present are included in the screen, and that other components present in the sample matrix are also considered, for example maize or soya bulking agents in foods. Finally, software that provides specificity screening will frequently report all of the possible matches. Careful appraisal of the degree of match reported should be undertaken before accepting an assay design. With the Primer Blast platform, in addition to performing primer and probe design, the software can also screen for cross-reactivity with non-targeted species. In the example illustrated on the current slide, the nucleotide database at NCBI has been selected as the reference source. Although this database is not curated and is redundant in terms of entries, it contains copies of all of the nucleotide sequences that have been submitted to GenBank, which is a requirement of most peer-reviewed journals for manuscript publication. It is also possible to specify which organism groups the primer sequences should be screened against. Finally, it is possible to specify the level of stringency that is to be used for comparison, for example, the number of matching bases between the query and a template. An example of the output from Primer Blast when performing an evaluation for primer specificity is shown on the current slide. Possible matches are shown in rank order of descending likelihood. Each record is shown in conjunction with a graphical representation of the relative position match between the template and the primer. The following slide summarizes the information provided by this e-seminar. In summary, commonly employed chemistries for quantitative PCR include double-stranded intercalating dyes such as cybergreen and evergreen, or dual-labeled hydrolysis probes such as TACMAN technology. When selecting an amplicon, regions of high GC, repetitive and palindromic sequences should be avoided. It is recommended that assay design be performed using appropriate software and the following design criteria be applied. 1. That primers should be between 20 to 24 bases in length with a melting temperature of approximately 60 degrees centigrade. 2. That probes should be between 20 to 30 bases in length with a melting temperature 7 to 10 degrees centigrade higher than the average for the primer pair. 3. That the 3' ends of primer pairs should not be complementary. 4. Any primer self-complementarity be avoided, and 5. That runs of 3 or more C's or G's at the 3' end of the primers should be avoided. Finally, it is recommended that assay specificity be evaluated in silico prior to having the primers and probes synthesized. If you would benefit from access to additional information, the following slide provides a short list of sources of information for designing quantitative PCR primers and probes. In addition to the publications listed here, the Gene Quantification website also provides a comprehensive set of links to additional sources of information that may be of interest. We would like to acknowledge the support received in preparing, delivering and funding this e-seminar, in particular, the Department for Environment, Food and Rural Affairs, the Government Chemist Programme 2017 to 2020, the Department for Business, Energy and Industrial Strategy, the Food Standards Agency and Food Standards Scotland. And finally, we would like to thank you for your attention.